In today's video, we will explore the fascinating world of molecular biology, focusing on the mechanisms that regulate gene expression. These processes determine cell function and can lead to diseases like cancer. We'll cover epigenetic regulation, transcription factors, chromatin remodeling, non-coding RNAs, DNA methylation, histone modifications, regulatory networks, RNA splicing, RNA editing, RNA localization, RNA stability, and post-transcriptional regulation. Let's get started. Epigenetic regulation involves heritable alterations in gene expression without changes in DNA sequence. Modifications like DNA methylation and histone modifications influence chromatin structure, and can be influenced by environmental factors. Dysregulation can contribute to disease development. Transcription factors are proteins that regulate gene expression by binding to specific DNA sequences. They can activate or inhibit gene expression by interacting with other molecules. Dysfunction of these proteins can lead to diseases. Chromatin remodeling is a process that changes the structure and composition of chromatin, the DNA and protein complex in chromosomes. Remodeling complexes use ATP energy to move or change nucleosomes, which affect gene expression and can contribute to disease. Non-coding RNAs, mRNAs, are RNA molecules that do not produce proteins but regulate gene expression. They can be small, microRNAs, small interfering RNAs, or long, interacting with mRNA or chromatin to control gene expression. Problems in mRNA regulation can lead to diseases like cancer. DNA methylation involves the addition of a methyl group to cytosine residues in DNA, typically occurring at CpG denucleotides. This epigenetic modification can lead to gene silencing and is crucial for normal development and cellular identity. DNA methyltransferases are enzymes responsible for adding methyl groups to DNA, and disruptions in their activity can result in abnormal methylation patterns and gene dysregulation. Altered DNA methylation patterns have been observed in various diseases, including imprinting disorders, neurodevelopmental disorders, and cancer. Histone modifications refer to various chemical modifications of histone proteins, including acetylation, methylation, phosphorylation, and ubiquitination. These modifications can dynamically alter the structure of chromatin and regulate gene expression. For example, acetylation of histones is generally associated with gene activation, while methylation can have both activating and repressive effects, depending on the specific lysine or arginine residues modified. Dysfunction in histone modifications can disrupt gene regulation and contribute to diseases, including hematological malignancies and autoimmune disorders. In cells, gene expression is controlled by regulatory networks involving transcription factors, chromatin regulators, and other molecules. These networks enable precise control of gene expression in response to environmental or developmental signals. Understanding these networks can help identify therapeutic targets for diseases. RNA splicing is a vital part of gene expression, removing unnecessary parts called introns from pre-mRNA and joining the remaining exons. Alternative splicing allows for different combinations of exons, resulting in multiple mRNA transcripts. Dysregulation of splicing can cause diseases like muscular dystrophies and neurodegenerative disorders. RNA editing is a post-transcriptional modification process that involves the alteration of RNA sequences, typically by the insertion, deletion, or modification of nucleotides. This process can be mediated by RNA editing enzymes, such as adenosine deaminases acting on RNA, ADARs, and can lead to changes in gene expression and protein function. RNA editing is particularly prevalent in the brain, where it can modulate neurotransmitter receptors and influence neuronal excitability. Dysregulation of RNA editing has been implicated in psychiatric disorders, such as schizophrenia, as well as neurodegenerative diseases. In addition to their coding potential, specific mRNAs can be selectively localized within cells, allowing for localized protein synthesis and spatial control of gene expression. This process is facilitated by RNA-binding proteins and RNA localization elements within the mRNA sequence. RNA localization is critical for various cellular processes, including embryonic development, neuronal connectivity, and immune responses. Dysregulation of RNA localization can disrupt normal cellular functions and contribute to diseases, including developmental disorders and certain cancers. RNA molecules have specific lifespans within cells and can be regulated through mechanisms that control their stability and degradation. Multiple factors, including mRNA sequence elements, RNA-binding proteins, and miRNAs, can influence the stability of RNA molecules. For example, miRNAs can bind to specific mRNA transcripts and promote their degradation or inhibit their translation. Dysregulation of RNA stability and degradation processes can result in abnormal gene expression patterns and contribute to various diseases, such as cardiovascular diseases and autoimmune disorders. Post-transcriptional regulation refers to the processes that occur after transcription, where RNA molecules undergo modifications, such as splicing, editing, and RNA decay, before they are utilized for protein synthesis. It also includes the regulation of mRNA transport, translation, and protein localization. These post-transcriptional processes allow for additional layers of control over gene expression and contribute to the fine-tuning of cellular functions. Dysfunction in post-transcriptional regulation can lead to a wide range of diseases, including neurological disorders and metabolic syndromes. Gene expression regulation is a complex process involving various factors like epigenetic modifications, transcription factors, chromatin remodeling, and non-coding RNAs. Understanding these mechanisms can aid in identifying disease targets for treatment and improving human health.